For our fourth talk, it's titled Genetic Algorithms for High-Dimensional Feature Refinement. The speaker is Kyle Covington, who's the director of bioinformatics at Castle. He brings with him over a decade of experience in the genomic space. He completed his PhD in translational medicine from Baylor College of Medicine. He's a scientist dedicated to advancing precision medicine through computational biology and genomic analysis. His research focuses on uncovering the molecular mechanisms driving cancer, leveraging large-scale genomic datasets to identify biomarkers, refine prognostic models, and enhance therapeutic strategies. With a strong foundation in oncology and bioinformatics, he has contributed to groundbreaking studies in cancer genomics, including pan-cancer analyses, mutational signature profiling, and machine learning applications in tumor classification. His work has been instrumental in developing predictive models for cancer progression and treatment response, and help bridge the gap between complex genomic data and clinical decision-making. Beyond research, he's passionate about collaboration and innovation. He thrives in interdisciplinary environments where computational methods intersect with clinical applications, driving forward discoveries that improve patient outcomes. Kyle, over to you.
Thank you so much. You couldn't hear me the first time I asked if you could hear me okay, because I was on mute. Thank you so much for inviting me to we can't see the audience as presenters if you don't know. So I assume this is a very well attended event, just given the number of questions. So again, thank you for inviting me. And I wanted to talk about, because it's a bioinformatics and bioinformatics data science type of group. It's a technique that I didn't necessarily invent this, but I tend to really like it for these cases where you have to move from high dimensional features to a smaller assay that you can deploy maybe without all of the things that the past set of presenters have been discussing, of moving around multi gigabyte data files. So what's the problem, right? High throughput transcriptomic and proteomic and metabolomic data sets, they generate thousands of features, and sometimes you want to reduce these features because your patent attorney says you can't patent the whole genome, or you want to move to some different platform that is more designed for high turnaround time. They want turnaround time to be a day, not 5 days. Or you want to use predictive algorithms that are suited to fewer features because some predictive algorithms don't really work very well with a high K, low N configuration. And how do you go about doing that, right? Like you can do feature selection, you can try to winnow down to a set of smaller set of features with good practices, but sometimes going from a 500 marker pathway viewpoint to, or metagene viewpoint, to something that you can fit on a PCR TLDA card, right, is pretty challenging. And that in itself is kind of a dual optimization problem, right? You have to optimize for panel size, so smaller panel size is favored, but also predictive accuracy, right? And genetic algorithms, I feel, are very useful for that. So this was formalized by John Holland in the 1970s, and the approach uses some key concepts. So one is that there have to be chromosomes. So there's markers. These are like the feature vectors that we would talk about in high throughput transcriptomics, right? They're the genes. So these are markers. These are chromosomes in case of genetic algorithms. There's a fitness function. So we want it to do something. So we can score different solutions to the problem, right? And we can say this solution is better than that solution. So we don't know that we might be able to make compromises on the number of markers, but we know too many is bad. So too many is just an outright failure. But maybe we could negotiate for smaller numbers if they were more fit. There's a selection process, and that's entirely dependent on the fitness function. So you use your fitness function to score everybody. You pick the prettiest things that are going forward, and you reproduce those. Crossover, this is how solutions combine together to generate new solutions. And then mutation, which is how you can reintroduce features into the model or into the population that maybe were winnowed out too early. Maybe they got an unlucky draw and they were bottlenecked out. So when I was first asked to like describe this, I used pictures of ducks and because like you can imagine ducks, right? But in this case, this is my farm label. So this is the Yellowbird Creek Restoration Farm Yellowbird, and we want to get a bird, right? We want to have the best bird. So how does a genetic algorithm work? Well, the bird needs different components, right? It needs to be able to eat. It needs to maybe have some tail feathers so it can signal to a mating partner. Eyes would be useful so it can see where it's going. Flight would be kind of good. A body to hold everything together. So early on at generation one, maybe we don't need to have the perfect bird. We just need to have something that's kind of bird-like. And we can find like, oh, these are two almost bird-like things, and they do most of the things that we need our bird to do. So let's combine them together and then do another round. Well, because of the inheritance, the next generation gets the features that its prior generation had, and it can mix them up a little bit. And so now we get things that are a little bit more bird-like. Well, these all have bodies. That's really good because that's a useful feature. And then more of them have beaks and eyes. And we keep doing this from generation to generation until finally, you get convergence where pretty much the selective pressure has been as extreme as it can be until you can get to something that's actually working. So now everything just looks like this beautiful yellow bird. So I like to use like examples that folks can follow. So let's set up in a little toy example, like how we're going to track this. So genetic algorithm example setup. We're going to create a 5,000 feature by 600 observation toy data set. The data set is going to be labeled A and B. We want to predict A and B ultimately. That's what we're going to get paid for, is can I tell the difference between A and B? Now, we're going to have a little game here where B has hidden states C, D, and E. So there's actually three subsets of B that are distinct from each other, but they all lead to this observable outcome B, right? This is very common in real biological processes where you'll have one set of mutations or one set of transcriptomic changes is leading to, high motility in the cell. Another one's leading to high survival in circulation. Another is leading to proliferation at different sites. All three of those may rely on very different underlying biological processes. They all contribute to increased metastatic potential. So you observe metastasis. You don't know why it happened. Well, it's combinations of those underlying states is why it happened in this world, right? So each of these leads to observable state B, and we want to have a model that has 30 or fewer predictors. That's our other constraint on this system. So we have no idea if we put this away, which of these 5,000 features are going to be useful. We're going to try to find them, right? So we're going to set our labels. There's A and B. There's the hidden state C, D, and E. And then we're going to spike in some real predictors, right? So we're going to spike in predictors 1 through 3 are decent at predicting state C because they have a different mean. Same thing for 4 through 6 and 7 through 9. These are really fun features. So these are ratio features. So the features between 10 and ultimately 21 is going to have 11 being essentially the ratio, some relative ratio of feature 10. So 11 is 11 divided by 10 is a really, really good predictor of states C and D. Not of A, not of E, but just C and D. We're doing that for D and E, and then E and C similarly. Now, when would you really run into something like this? Well, this is kind of exactly what you have if you have a good marker, like a good control marker for cell type, which is your tumor cell type, and then overexpression of another marker, and you then layer on contamination from something else, right? Like you have stromal contamination. Well, you do your RNA-seq, you do RPM normalization, you get exactly this, and it's scaled by the stromal contamination. We're also going to make features 30 through 40 just generally good predictors of, they have like an offset of plus or minus 0.1 from the mean for those features just of states A and B. We're going to split that into a train, a test, and a validation set. Now, I wrote down always, always, always do this because genetic algorithms, like evolution, is the most powerful generic optimizer that I think we know about. Someone can fact check me on that, but I don't think anyone knows of a more powerful generic optimizer. Um, this thing will lie to you. So you need to be very careful with it. So always have a test data set to compare against your train, use that for the fitness function, and then keep another set aside for validation. Don't these models will absolutely optimize to be good predictors of the test data set, whatever you're optimizing them towards. So you have to keep something aside and use that for your validation. And then we're just going to do the usual pre-processing center and scale on our train test and validation set. So I can already hear someone in the audience about to get up saying, well, Kyle, why can't I just use Elastic Net? Like Elastic Net has to work. It always like, and I agree with you. I want Elastic Net to work as well. It always seems so beautiful, this idea that I can just, oh, I can do feature reduction and model fitting at the same time. Let's try it out. It doesn't work. Like just in this case, if we just do GLM net and we run it against our trained data set, the accuracy is just guessing. So, well, why didn't that work? Well, it's just not exactly the right thing for this particular problem, right? Because the best predictors of state were those ratio features, and they're kind of hard to solve. And the difference in the means and with the random selection that we have, it wasn't of the other markers, just wasn't quite strong enough to get really good prediction out of it. And so, the logistic regression methods just tend not to work if you have multi-state problems. And trees can handle multi-state really well, but they don't handle ratios. So maybe we could go do like C50. I could run C50, but that might work really well. Actually, I haven't tried it, but we could run C50 and see if that, how that works. But neural networks, these are really excellent generic learners. Like this is exactly the type of thing that you want, right? This is the best generic learner that we have. And it's capable of actually recognizing multiple states. So you can have, these layered neurons that are firing because they see, oh, this one has a high ratio, right, of these two markers in a specific state. It recognizes that. Maybe it doesn't recognize the others, but you can then layer that together and you can do a sum and you can say, oh, okay, I see now there's these three different things that are going on, and that's going to predict my outcome. And if you do that on the best case scenario with just generic neural nets, these are all default settings. I'm just running default settings for everything. It that works really well. Actually, the regression model seems to work really well in these 40 key predictors. It got the answer right. Actually, it is a three-state problem, and that's the one with the highest accuracy and highest kappa. So neural nets would be really good if but only we could give it a small enough data set so that it could actually do the solving. So now we're going to combine these ideas, right? So we're going to build a population of candidate solutions. Just believe me that 3 times 5,000 is the same as 500 times 30. So we're going to make 500 different groups of 30 marker sets, 500 sets with 30 markers in each set. And we're just gonna, so we're gonna initialize that. We're gonna have all of our markers. These are gonna be the birds that we're going to test. We're gonna see which one is the prettiest bird. And we're gonna run this through a fit function. So we're gonna make that neural net fit on the marker list that we're giving it. We're gonna run predict against the test. That's gonna be our fitness function. And then we're gonna just return that. Now, if we do that, it tends not to work very well. And that makes sense because on average, like none of these predictors are really very good. Um, but some of them are. Some of them are actually okay. So we can just keep doing this. We'll just select the best fits. We'll mate them together and we'll keep going. So if you want to do this, we're going to order by the fit scores. we're just going to test against the validation, the testing data, and then score fitness. In this case, it's just going to be kappa. Take the top 125, and then we're going to randomly assign these to each other and run this mate function. This is going to handle the crossing over, right? Because we want to combine features from the best solutions together. In this particular case, I'm just concatenating marker list 1 and marker list 2, taking 27. Kyle, why not 30? Well, that's where the mutation comes in. So we take 27, and then we select 3 from the remaining training data set, the remaining features that are not in the ones that we picked. So this is how we're going to reintroduce uh, markers that we had previously dropped out back into the data set. This is all configurable, right? You can change, you can increase the mutation rate. You can use some other ideas are like using variable importance to pick these, not just blindly picking them. But in this case, we're just going to do some simple stuff, right? Because we got work to do. So we're going to just do that. And that's how we're going to make the next generation of useful models. And if we do that until it converges, and I think this converged after about 6 iterations. One, we can take these surviving that the rank ordered surviving fits, right? So these would be the top 200, 125 solutions. And we're going to just plot those against our predictive markers. And look at this. It found 18 and 19, and it was starting to find 10 and 11. 10 and 11 were starting to become pretty common. And if we also just run through these and we say, well, sum the variable importance and do a count. So you can see that 18 and 19 were the top predictors by the sum of their variable importance. They were in every single surviving model. And then 10 and 11 was again starting to get picked up. This was going to start propagating through, and they were also very high. After that, it's these markers that are sort of like in mutation analysis you call them like passenger mutations. These are just passenger features that happen to coincide with some of the highly predictive family members, high predictive markers, chromosomes, and they keep getting put back in just by luck because they're in a good model. Um, so the performance of that top model, it was not quite as accurate as the best model that used all 40 features, but it was still passable, right? Like this, this would probably make a good diagnostic assay for you. And it was picking basically two of the states that would recognize all of the hidden states, right? So this one features 10 and 11 would recognize states C and D, and 18 and 19 we're going to recognize states E and C. So some other like tips and tricks that you can do from this is one, just now you get to kind of play God, right? Like you get to direct evolution. So you can use variable importance just to spike in highly predictive variables into new models. So if we do that and we spike in 10 and 11 and 18 and 19, you'll see that you get this sort of dominating evolution pattern where it just wants to see 10, 11, 18, and 19. They're in every single model and it doesn't find anything else. So this is kind of like if you imagine, it's very difficult to turn a bird wing into a bat wing. If you look at the skeletal structure, because a bat wing uses its little fingers and the bird has just merged all of their metatarsals together. It's not going to go the other way. So another thing you can do is you can remove markers with high variable importance and see if anything else comes out. And if we do that, we remove 10, 11, 18, and 19, and we run, it finds 20 and 21. So now, it didn't get the inertia of the bird wing. Now it picked a bat wing that it can fly with, by analogy. So you can kind of keep doing that. You can augment the mutation rate. You can change the selection pressure to get higher selection pressure. You can use larger models than you want. Like let's say you targeted 30, but maybe we just want to run 60 markers at a time, and we're just going to prune later, right? So we can do that. You can combine predictive markers if you have maybe two very distinct sets of predictive markers, like you're finding two, very distinct marker sets when you do the pruning step. Well, you could combine those into ensemble models, right? Like, why not do that? And have whole families of solutions that are relying on maybe a smaller set of genes that you can run on commodity hardware. Um, So if we then take that the performance in the validation data set, again, always compare to validation because this thing absolutely will lie to you. And we take 10, 11, 18, 19, 20, and 21, and we just build a model on that. This is our best genetic algorithm derived neural network, and then this was ideal, which has all 40 in it. And it all 40, to be fair, if we were prescient and we could pick out the 40 markers out of our, you know, set of 5,000, transcriptomic features, that would be really cool if we could do that. But, I feel like this solution got pretty close to what we would consider the right answer. It's highly predictive, much more predictive than, the regression models. And, now it fits nicely into a nice, commodity card that you could order PCR primers for. Um, so trying to catch us back up on time here. Oh, good. I think we will finish on time. Just everyone will have like an hour to go move their cars out of the parking lot. So in summary, genetic algorithms can be efficient ways to screen otherwise very large feature spaces. You don't have to visit every possible iterative combination. You can sort of like select and then move into something that's going to be a passable solution. They allow the use of model families which otherwise struggle with large feature spaces. And I think I've said it 10 times in the talk. They are powerful, very powerful optimizers. Always use a separate test and validation set to limit overfitting. And thank you for your time and attention. I'd love to answer any questions.
Hey Kyle, great talk. That was really, really cool and got me thinking. On your last note about overfitting, what are some of the learnings that you saw creating this model using a neural net? For the folks that don't know, neural nets, although are powerful, they do tend to make some interesting calls and overfit.
Yeah, well, one of the one of the first times I ever used this is I said to myself, "Oh, I wonder what would happen if I just shuffle the labels." And it gave me a fantastic answer. Like it was incredibly predictive because you're in this approach, right? You're optimizing to your validation set. So this is incredibly prone to overfitting, and that's why it's somewhat dangerous. I'm kind of giving you all a dangerous tool if you go to try it. So that's why I keep emphasizing, always have another validation set. But neural nets, I agree with you, also can easily overfit. So the only way I've found out to combat that is split up your validation. Make sure you're only go into the validation set one time. You have to be convinced I love this model. This is what I'm going to go into production with. Now I'm going to try it in validation and be willing to say, okay, if that doesn't work, I scrap it.
Cool, thank you.
It's hard to convince executive team of that sometimes, but you got to be disciplined with it.
You simulated annealing a while back, and I remember that there was this interesting feature it had where you could jump out of these local optima by introducing a step akin to something that happens in physics where things just jump out of their favorite states. And you mentioned something about when you converge to a bird wing, you can't really converge to a bat wing, etc. I was just curious, is there a jumping step in GA that's analogous or potentially, I guess you could introduce it?
Well, yeah, that's what I was introducing by removing the features that the first iteration tended to like the best, right?
Oh, that's
So
how it does it. Right, right. I
yeah,
see.
so that's how I do it is, if you're getting something that's giving you like a really, really strong solution, it it's hard, right? Because, in at least in the case of, let's say, picking features 20 and 21, um, it's the chances are, what is it? There's a 30 in 5,000 chance that it'll get marker 20, and then another 30 in 5,000 that it finds 21. So you have to get
Yeah.
lucky that it has to get both of them together, right, at the same
Yeah.
time. And if getting 20 or 21 was kind of useful at first, then that's more likely to get retained and have a chance
Yes.
of finding its useful partner, right? But if it's staring in the face of this big sun, which is 18 and 19 and 10 and 11, which are really good predictors by themselves, the addition of just getting one of them, that's the example of the bird, right? Like, so if you have a population bottleneck, and you have a bunch of organisms that aren't really well fit to their task necessarily, but they're fit enough. They're doing a good enough job that they can make it through. Then they'll spread, and GAs are very similar to that.
I think there's a random part to that also in simulated. I'm sorry, I'm probably
Yeah.
misremembering, but in your case, I think you deterministically removed the best ones. I guess you would want to also do some random starts, and I mean, not necessarily the best ones, but just any ones, right? I mean, I guess you could do that.
Oh, sure. Yeah. Like I said, you can random start again. You can
Right. Yes.
augmenting mutation rate is another way of if you're just trying to get lucky to get good features. I mean, you have to somewhat believe that there's a good feature, at least there's good features in there, right? Like the data
Right.
is somewhat predictive, yeah. Um, Now, how predictive you may not know, but you'd have to go in with the belief that there's something there to find, and then you want to just sort of squeeze everything you can out of it. So I find variable importance to be really useful for that. Both on the optimization side, and then trying to then removing those and trying to fish out other secondary features that maybe just got an unlucky draw the first time. But yeah, random seeding is good too. Yeah, you can randomly seed and I this whole thing on 5,000 features with 600 things ran like overnight on a 5-core machine. So, yeah, in real life, devote a little bit more effort to it than what we gave to this toy data set.
Hi, Kyle. Nice talk.
Thanks.
I was wondering, like, compared to like the genetic algorithms, have you tried just regular algorithms like random forest or XGBoost to see how they can filter the critical features out? Because say random forest can do the sampling and build different trees and then score the ones that show the concurrency across multiple models and stuff like that.
Yeah, I've found random forests, I mean that's you can tune them to be reductive, right, to a set that you'd be interested in. I don't know uh that's one thing I haven't really tried it. The talk was about GAs, right? Because we all know about random forest. So I have tried random forest, but I find the trees, right? Trees are going to make decisions on cut points, right? And so I guess that was the one thing I was pointing out with these. Uh, uh, sometimes you do have things that are clear cut points, right? Like just, less than this marker, less than this value is going to be a good, as a predictive thing. Um, but when you're layering on other things that happen in real biological systems, you have tumor contamination, right? So I work primarily oncology, right? So, it's stromal contamination. And now you have, stromal RNA kind of competing with this signal that you're getting from another tissue type. So I feel like that the neuronal nets, just in my experience, tend to work better than random forests, and I think it's because of that. Um, Whereas the forests essentially have to make like thousands of cut point rules, right? Like if this, then this, and if this and this, then predict, the outcome, right? It has to step across the gradient. It can't just impute it.
Yeah, thanks for the talk, Kyle. So from and maybe correct me if I'm wrong, but I think one of the points you're trying to make is that the genetic algorithm with a neural network is running with slightly better complexity than if you were to have a neural network with the 40 predictors. Am I understanding that correctly, or does the complexity not change between the two?
Uh, well, but you have to screen down, right, to the to pick your markers, right? Like you don't know if you're doing the experiment in real life which of the 5,000 markers are going to be useful, right? So the neural nets I haven't found fit very well. Like they tend to really overfit if you give them that many a high K low N problem. There's just too many free parameters, and the neural net will just make that was, I guess, the first person's point with the question, right? Is that the neural nets tend to over optimize with big K small n problems. So this is kind of cheating. Like I get to feed the big K, but in small little bites to the neural networks and see which one is performing the best. If that's answering your question.
Announcement. Thank you, Kyle.
Yeah.
Okay. Thanks so much.
Thank you so much.
Thank our speakers again. Yeah.